The Bioreaction-Variation Network, or BVN, is a new AI model that infers molecular and physiological parameter networks from biological experimental data. It is designed to be implemented at the laboratory scale with a customizable, context-based framework. In this presentation, I will introduce what BVN can do, from network construction to inference, and show an example of its analytical results. BVN is built from two types of nodes, model and target. Each group, consisting of a model and its connected targets, is extracted from a single research article. Model and target features include contextual information, such as species, model details, and biosample types for the model features, and molecular types, analytical methods, and the direction of change for the target features. All of these are encoded as BioBERT-based embeddings, forming a small subgraph that represents one article within the overall network. Targets are interconnected with each other within each article. Then the same process is repeated for another paper, extracting related text segments to generate additional subgraphs. As more papers are processed, the BVN gradually expands, linking together biological findings reported across multiple studies. These subgraphs are connected through hub nodes. A hub node integrates multiple features, representing alternative physiological states that are influenced by different experimental models. Through these hub nodes, the BVN unifies scattered findings into a continuous biological network. When an experimental model identical to an existing one is added, its new features are integrated into the same model or target node. By collecting these varied features within a single node, BVN captures the biological diversity and variability underlying similar experimental conditions. When targets connected to new experimental models are introduced, BVN expands by adding multiple and sometimes competing edges. This continual addition of diverse relationships allows BVN to represent the competitive and cooperative dynamics found in real biological systems. The currently released BVN was trained under the assumption that all physiological stimuli are simultaneously active. This so-called full activation mode enables all biological factors to interact. When the network is activated as a whole, the resulting node features after message passing can be interpreted as each node's inherent position and role within the biological system. In the first learning phase, BVN learns the model-to-target relationships using a multi-head graph attention convolution mechanism. This allows the model to determine how strongly each target node is influenced by a given experimental model, represented as an attention weight. Next, BVN learns how the targets themselves are related. This process proceeds in two steps. The first step captures local structural dependencies through message passing between neighboring target nodes. During this process, the original node features from the dataset serve as teacher signals, helping BVN reconstruct the structure of local subgraphs. The second step focuses on target-to-target directionality, learned through what we call pairwise domination. Here, domination describes the upstream-downstream relationship between two directly connected target nodes. For example, if the input feature from node A is similar to the average of input features received by node B, node A is considered dominant over node B, meaning that node A functions as the upstream regulator in that pair. BVN calculates this dominance across all possible target node pairs in the dataset, using it as a teacher signal to refine the directional structure of the network. A pre-trained BVN consists of the learned network of target nodes and the experimental models linked to them. Users can perform network inference by inputting their own experimental data, specifying the model conditions and the measured parameters with their changes. Inference begins by matching experimental models in BVN that are similar to the user's setup. Users can also define how many models to include. Typically, selecting about 5 matching models connects to over 10 target nodes, which then serve as the starting points for inference. The selected experimental models are used as start nodes for inference. The primary targets directly linked to these models are then identified. Users also provide their own results for specific targets, which serve as goal nodes. BVN then searches for all possible paths from each primary target to each goal. following the learned directionality of the target-to-target edges. For each explored path, BVN calculates message passing using the original edge attributes from the dataset. Among multiple possible edges connecting the same targets, BVN selects the optimal one whose features most closely match the input-output relationship. Message passing begins with updating the primary target's feature according to its attention weight, simulating the biological effect when that target is stimulated by the experimental model. This process is repeated for subsequent nodes, propagating messages step by step through the network. This message passing process is repeated across all explored paths. Finally, BVN evaluates every possible path combination to find the one that produces the mean goal node feature most similar to the user's input. In this way, BVN reconstructs intermediate networks consistent with previously reported data. A major advantage of the full activation mode is that BVN can perform this reconstruction directly from intact datasets, without any rewriting. The typical output of BVN inference is a network diagram. Here is an example generated from real input data. Each node and edge represents a molecular or physiological relationship. A node that has many inputs but no outputs is typically a goal node, and the similarity between the predicted and actual goal features can be quantified by the Euclidean distance. Nodes with multiple input and output edges often act as hub nodes, bridging different subgraphs. Further structural analysis, such as centrality assessment, can provide deeper insight into network organization. As demonstrated in this presentation, BVN is a novel AI framework that infers intermediate biological networks directly from experimental data. It can even perform inference at the individual level, making it especially powerful for classifying interindividual variation. For more information on the BVN architecture and detailed metrics, please refer to our publication.